topic discussion, the book of Cellular and Molecular Immunology. Today we are in chapter 5, where we'll discuss about antibodies and antigens. So first of all, we'll discuss this antibody structure. We'll discuss synthesis, assembly, and expression of immunoglobulin molecules. We'll discuss antibody binding of antigen. We'll discuss structure, function, relationship in antibody molecules. There are features related to antigen recognition, features related to effector function, and furthermore we will summarize. So let us start. Antibodies are circulating proteins that are produced in vertebrates in response to exposure to foreign structure known as antigen, and are the mediator of humoral immunity against all classes of microbes. Antibodies are extremely diverse and specific in their ability to recognize foreign molecules structure. Because these proteins were discovered as circulating protein that provided protection against diphtheria toxin, they were initially called the antitoxin. When it was appreciated that similar protein could be generated against many substances, not just the microbial toxin, they were given the general name antibodies. The substance that stimulate production of or were recognized by antibody were called antigen. Antibody and T-cell antigen receptor are the two classes of molecule used by adaptive immune system to specifically recognize and respond to antigen. Major histocompatibility complex MHC molecule also bind peptide antigen, but their specificity is very different and their function is to passively display peptide to T cell antigen receptor and not to respond to antigen in the way that antibody on B lymphocyte and T lymphocyte T cell receptor or on T lymphocyte do. Antibody were discovered almost a century before antigen receptor were identified on the T cell, and in comparison to T cell receptor and MHC molecules, antibody recognize a wider range of antigenic structure, have a greater ability to discriminate among different antigen, and bind antigen with a greatest strength. In this chapter, we will describe the structure and antigen binding properties of antibodies. Antibodies are synthesized only by cell of the B lymphocyte lineage, and they exist in two forms. This is membrane-bound antibodies on the surface of B lymphocyte functions as antigen receptor, and secreted antibody function to protect against the microbes. The recognition of antigen by membrane-bound antibodies on naive B cell activates these lymphocytes and initiate a humoral immune response. The activated B cells differentiate into plasma cells that secrete antibodies of the same specificity as antigen receptor. Secretory form of antibody are present in the plasma, the fluid portion of the blood, in mucosa secretion, and in interstitial fluid of the tissue. Antibodies recognize antigen and also have effector function that contribute to the elimination of antigen. Secreted antibodies neutralize microbial toxin, prevent the entry and the spread of pathogens, and trigger several effector mechanisms that eliminate the microbes. As we will discuss later, different regions of the antibody molecules are responsible for recognition of antigen and many of the effector function, but all function of antibody are initiated by specific antigen recognition. The elimination of antigen often requires interaction of antibody with other components of the immune system, including molecules such as a complement protein and cells such as phagocytes and mast cell. Antibody-mediated effector function include neutralization of microbes or toxin microbial products, activation of the complement system, opsonization of pathogen for enhanced phagocytosis, antibody-dependent cell-mediated cytotoxicity, by which antibody target infected cell for lysis by cell, of the innate immune system and antibody-mediated mast cell activation to expel parasitic worms. We will describe this function of antibodies in detail in chapter 13. When blood or plasma removed from an individual and placed in tube forms a clot, antibodies remain in residue fluid, which is called serum. Serum lack coagulation factors, which are consumed during clot formation, but contain all other protein found in plasma. Any serum sample that contains detectable antibody molecule that bind to particular antigen is commonly called an anti-serum. The study of antibodies and their reaction with antigen is therefore called serology. The concentration of antibody molecules in serum specifically for a particular antigen is often estimated by determining how many serial dilution of the serum can be made before binding to the antigen can no longer be detected. This process of determining the concentration of any substance by testing serial dilution is called titration, and the antibody concentration determined by this method is titer. The more dilution that are required, the higher is titer of the antibody molecule specific for particular antigen. So we have the features, table 5.1, features of antigen binding by antigen receptor, the features immunoglobulin IG, T cell receptor. So as we look for antigen binding, these are made up of 3 CDRs, the IG, T cell receptor made up of 3 CDRs. You can see antigen binding site. You see they are combined together with VH and 3 CDRs together with VL domain. But for T cell receptor is V E and 3 CDR in VBM domains in most common form of TCR. So nature of antigen that may be found actually macromolecular protein lipid polysaccharides in immunoglobulin, but in T cells we find peptide MHC complex. Nature of antigenic determinants recognized. These mostly in immunoglobulin they are linear and conformational determinants of various macromolecular and chemical, but in a T cell receptor, linear determinants of peptides, only few amino acid residue and peptide bond to the MHC. You will see them later. Affinity, the KD is 10 power negative 7 to 10 power negative 11. Average affinity for IgGs increasing during immune response, but for T cell is 10 power 5 to 10 power 7 negative. And on rate and off rate, the rapid on rate, variable of rate, slow on rate, slow off rate. So that's what is happening. And if you want to know what were the letters up there, the CDR means complementarity determining region, KD is dissociation constant, MHC is the major histocompatibility complex, MH is variable domain of heavy chain, and VL is variable domain of light chain, and V and VB variable domain of T cell receptor alpha and beta chains. So we have the V alpha and V beta that you have seen there. The structure and function of T cell molecule are discussed further in chapter 7. A healthy 70 kg adult human produces about 2 to 3 gram of antibodies every day. This is 2 to 3 gram of antibodies every day are produced by 70 kg person. Almost 2/3 of this is a type of antibody called immunoglobulin A, IgA, the 2/3, most of which is produced by intestinal plasma cell and secreted into the gut lumen. The antibody structure. An understanding of the structure of antibody has provided important insight into their function. The analysis of antibody structure also laid the foundation of elucidating the mechanism of antigen receptor diversity that we will consider in depth in chapter 8. Early studies of antibody structures relied on antibody purified from the blood of an individual immunized with various antigen. It was not possible using this approach to define antibody structure precisely because serum contains a mixture of different antibodies produced by many clones of B lymphocytes that may each bind to different portion epitope of antigen. These antibody mixtures are called the polyclonal antibodies. A major breakthrough in obtaining antibody whose structure could be elucidated was at the discovery of that patient with multiple myeloma. A monoclonal tumor of antibody-producing plasma cell often have a large amount of biochemically identical antibody molecule produced by neoplastic clones in their blood and urine. Immunologists found that these antibodies could be purified to homogeneity and analyzed. The realization that myeloma cells make one type of immunoglobulin led to the development of technology to produce a monoclonal antibody described later in the chapter. The availability of homogeneous population of antibody and monoclonal antibody producing plasma cell facilitate the detailed structure analysis of antibody molecule and molecular cloning of genes of an individual antibodies. These were the important advance in our understanding of adaptive immune system. General features of antibody structure. Plasma or serum proteins can be physically separated based on the solubility characteristics into albumin and globulin and may be more precisely separated based on difference in charge using a technique called electrophoresis. In electrophoretic separation of serum or plasma, most antibodies are found in a third fastest migrating group of globulin, namely gamma globulin for the third letter of Greek alphabet. Note that gamma globulin include all classes of antibody described later, not just IgG class. Another common name for antibody is immunoglobulin, Ig, referring to the immune-conferred portion of the globulin fraction of serum or plasma. The term immunoglobulin and antibodies are used interchangeably throughout this book. All antibody molecules share the same basic structural characteristics, but display remarkable variability in the region that bind antigen. This variability of antigen binding region accounts for the capacity of different antibodies to bind a tremendous number of structurally diverse antigen. In every individual, there are millions of different clones of B cell, each producing antibody molecule with identical antigen binding site, but that differ from antigen binding site of antibody produced by other clones. The effector function and common physicochemical properties of antibody are associated with the non-antigen binding portion, which exhibit relatively few variation among different antibodies. So the structure of antibody molecules, the schematic diagram of secreted immunoglobulin, you see here, this is it is like Y shape. It is having the heavy chain. These are in. There is a light chain outside. There is a Fab region. There is FC region, and there is FC receptor complement. But here again there is tail piece. Here there is hinge whereby they come together. So they can come in the plasma membrane of the B cells, and they are produced. They say this is heavy chain, and in here these are binding antigen binding site, and it is combined together, working together. This with the sulfide bond tail piece. We will see them as we are seeing it. That help you to just remember. So as we say, this is the schematic diagram of secreted immunoglobulin, IgG molecule. The antigen binding site are formed by juxtaposition of variable of the light chain variable and variable heavy chain domain. The heavy chain C region and the end in tail pieces. The location of complement binding and FC receptor binding site within a heavy chain constant region are approximation. B, this is schematic diagram of membrane bound IgM. You see this is IgM, this was IgG, and the IgM molecule has one more constant heavy chain that IgG has, and the membrane from the antibody has C-terminal transmembrane and cytoplasmic portion that anchor the molecule to plasma membrane. C, the structure of human IgG molecule has revealed by X-ray chromatography, this one. So if you do the X-ray chromatography, this is how you're going to see it. And in this ribbon diagram of secreted IgG molecule, the identical heavy chains are colored blue and red so that they can easily visualized. Although they are identical and the light chain are colored green, carbohydrates that are covalently bound to IgM heavy chain protein are shown in gray. So an antibody molecule has a symmetric core structure composed of two identical light chain and two identical heavy chain, as you saw it in figure 5.1. Both the light chain and heavy chain contain a series of repeated homologous structure unity, each about 110 amino acid residue in length, that fold independently in a globular motif that is called the Ig domain, which we introduced in chapter 3 and chapter 4. An Ig domain contains two layers of beta plated sheet, each layer composed of 3 to 5 strands of antiparallel polypeptide chain. The two layers are held together by disulfide bridge, and adjacent strands of each beta sheet are connected by short loops. It is the amino acid in some of these loops that are the most variable and critical for antigen recognition, as discussed later in the chapter. You'll see the figure 2.5.2. As we see, these are strands, they look like plates, right? This is the structure of an immunoglobulin domain. Each domain is composed of two antiparallel array of beta strand, colored yellow and red. These are yellow and the red one, to form two pleated sheets called together by the sulfide bond. So these one are sheets called together by the sulfide bond. The diagram shows an immunoglobulin constant C. It's the C region domain containing 3 and 4 beta strands in the two adjacent sheets. N and C at the end of the polypeptide chain refer to amino and carboxyl terminal respectively. Therefore, this is amino, and there is C, carboxyl terminal. Antibody heavy chain and light chain both consist of amino terminal variable V region that participate in antigen recognition and carboxyl terminal constant C region. The C region of the heavy chain help mediate some of the effector function of the antibodies. In the heavy chain, the V region is composed of one Ig domain, and C region is composed of 3 or 4 Ig domain. Each light chain is composed of one V region, variable region, Ig domain, and one C constant region, Ig domain. Variable regions are so named because their amino acid sequence vary among antibodies made by different B cell clones. The variable region of one heavy chain and the adjacent variable region of one light chain form an antigen binding site. We'll see it we already saw it in figure 5.1. Because the core structure unit of each antibody molecules contain two heavy chains and two light chains, every antibody molecule has at least two antigen binding sites. C region Ig domain are spatially separated from the antigen binding site and do not participate in antigen recognition. The heavy chain C region interact with other molecules and cell of the immune system and therefore help mediate most of the effector function of the antibodies. In addition, heavy chain exists in two forms that differ at their carboxyl terminal ends. One form of the heavy chain anchor membrane-bound antibodies in the plasma membrane of the B lymphocytes, and the other form is bound only in secreted antibodies. The constant region of the light chain do not participate in effector function and are not directly anchored to the cell membrane. Heavy and light chain are covalently linked by disulfide bond formed between a cysteine residue in carboxyl terminus of the light chain and the constant heavy chain domain of heavy chain. Non-covalent interaction between the L light chain and heavy chain domain, variable light chain and variable heavy chain domains, and between the constant light and constant heavy chain domain may also contribute to the association of heavy and light chains. Two heavy chains of each antibody molecules are covalently linked by the sulfide bond. There are different kinds of antibody called classes or isotopes that have different heavy chain structure. Discussed in detail later in the chapter, in the IgG isotope, the disulfide bond are formed between cysteine residue in the C constant heavy chain 2 domain close to an unfolded segment called the Hing region, which connect the constant heavy chain 1 domain to constant heavy chain 2 region. This segment is described in more detail later in the chapter. In other isotopes, the sulfide bond may be in different location. Non-covalent interaction, e.g. Between the third constant heavy chain domain, also contribute to the heavy chain pairing. Antigen binding portion of antibody molecule is the Fab region, and C terminal end that is involved in effector function is this Fc region. So, these regions were originally identified by proteolysis of rabbit IgG molecules. In this molecule, the hang region between the constant heavy chain 1 and constant heavy chain 2 domain of the heavy chain is the segment of most susceptible to proteolytic cleavage because this region is unfolded and proteolytic sites are thus easily accessible. If rabbit IgG is treated with enzyme papain under a condition of limited proteolysis, the enzyme acts on the hing region and cleave the IgG into 3 separate pieces. 2 of the pieces are identical to each other and consist of the complete light chain, variable light chain, and constant light chain. Also associated with the variable heavy chain and variable and the constant heavy chain fragment of the heavy chain. These fragments retain the ability to bind antigen because each contain paired variable light chain, light and variable heavy domain, and they are called Fab, the fragment antigen binding. The third piece is composed of 2 identical disulfide linked peptides, each containing a heavy chain, the constant heavy chain 2 and constant heavy chain 3 domains. These pieces of IgG has a property of self-associate to a crystallized into the lattice and is therefore called Fc, fragment crystallizable. When pepsin instead of papain is used to cleave the rabbit IgG under limiting conditions, the proteolytic occur in distal to the hang gene, generating a Fab2 fragment of IgG with the hing and interchain disulfide bond intact and 2 identical antigen binding sites. The basic organization of antibody molecule deducted from the rabbit IgG proteolysis experiment is common to all IgG molecules of all classes and all species, and the term Fab, F-A-B, to the bracket you see, and Fc are widely used to describe these different portions of antibodies. In fact, this experiment provided the first evidence that antigen recognition function and the effector function of IgG molecule are spatially spatially separated. Many other proteins in the immune system, as well as numerous proteins with no known immunological function, contain domain with Ig fold structure, that is, two adjacent beta-pleated sheets held together by the sulfide bridge. All molecules that contain this type of domain are said to belong to Ig superfamily, and all gene segments encoded the Ig domain of these molecules are thought to have evolved from one ancestral genes. Ig domains are classified as V-like or C-like on the basis of closest homology to either IgV or IgC domains. V domains are formed from a longer polypeptide chain than C domains and contain two extra beta-stranded strands with beta sheets and sandwich. Some members of Ig superfamily were described in Chapter 3, endothelial adhesion molecule, ICAM1, and the VCAM1. And Chapter 4, the natural killer cells, Krieger receptor. Example of Ig superfamily members of relevance in immune system are depicted in Figure 5.4. So here we have the Figure 5.3. We have A, B. Then in the A we have, you see the heavy chain, the light chain. When the papain is cut, it cut these two, separated this to three. You get the structures you see here. You have fab. Then papain product is here. Then this is by papain. But when you use pepsin, it cuts here and leaves the hinge joint here. That's why you see the hinge joint here. So the peptide fragment, as you see, so proteolytic fragment of immunoglobulin, IgG molecule. L studies of properties of proteolytic fragments of rabbit Ig provided key insight into general structure features of all Ig molecule. Rabbit Ig molecules are cleaved by enzyme papain, A, you see, and pepsin in B. And they are at site indicated by arrows. Papain digest allow separation of two antigen binding region, the Fab fragment from the portion of IgG molecule that bind to the complement, and the Fc receptor fragment. Pepsin generates the single bivalent antigen binding fragments, and C at the end of the peptide chain refer to the amino acid copy receptor respectively. Structural features of antibody variable region. So most of the sequence differences and variability among different antibody are confined to three short stretches in V region of the heavy chain and to three stretches in V region of the light chain. These are segments of the greatest diversity. These segments of the greatest diversity are known as hypervariable regions. They correspond to three protruding loops connecting adjacent strands of the beta sheets that make up the V domains of Ig heavy and light chain proteins. The hypervariable regions are each about 10 amino acid residue long, and they are held in place by more conserved framework sequence that make up the Ig domain of V region. Because these sequences form a surface that is complementary to three-dimensional shape of the bound antigen, the hypervariable region are also called the complementarity determining region, CDRS. Proceeding from either the variable light or the variable heavy amino terminus, these regions are called CDR1, CDR2. This is complementarity determining region 1, complementarity determining region 2, and complementarity determining region 3. Sequence difference among the complementarity determining region of different antibody molecule contribute to distinct interaction surface and therefore to specificity of individual antibodies. The complementarity determining region 3s of both the the variable heavy chain segment and variable light chain segment are the most variable for complementarity determining regions. As we will discuss in Chapter 8, there are special mechanisms for generating more sequence of diversity in complementarity determining region 3 than in complementarity determining region 2 and 1 and 2. In the antibody molecule, the three hypervariable region of variable light domain and the three hypervariable region of variable heavy chain domain are brought together to create an antigen binding surface. Hypervariable loops can be thought to resemble finger protruding from each variable domains, with 3 fingers from the heavy chain and 3 fingers from light chain coming together to form an antigen binding site. The ability of variable regions to fold into an Ig domain is mostly determined by a conserved sequence of framework regions adjacent to the complementarity determining regions. Confined of the sequence of variable to 3 short stretches allow the basic structure of all antibody to be maintained despite the variability that contribute to the specificity of different antibodies. So you see, this is the structure. Example of immunoglobulin, superfamily protein in the immune system. Shown here are the membrane bound Ig molecule, IgG. You see the structure. Here we have, these are structure, they are bound in the membrane. The T receptor, major histocompatibility complex, class 1 molecule, the CD4 co-receptor of T. So these are all structure that you can see. Crystallography analysis of antigen-antibody complexes show that amino acid residue of complementarity determining region form multiple contact with bound antigen. The most extensive contact is with the third hypervariable region. However, antigen binding is not solely the function of complementarity determining regions, and the framework residue also may contact the antigen. Moreover, in the binding of some antigen by a specific antibody molecule, one or more of complementary determining regions may be outside the region of contact with the antigen. Therefore, binding to certain antigen may occur independently of some of the Ig heavy or light chain. So these are just structure again. The hypervariable region in immunoglobulin, A and B. There is A and B. Vertical lines depict the extent of variable. They have the variability. You study more, and we see this is the structure again. Binding of antigen by antibody. Schematic review of complementary determining region. Generating an antigen binding site, as we are told. So these are the ones that are making that. So we see the antigen binding site. Yeah. So let us discuss the structural features of antibody constant region. Antibody molecules can be divided into distinct classes and subclasses on basis of difference in the structure of their heavy chain region, the C region, the heavy chain constant region. The different antibody molecule are classified molecule are called classes or isotopes and are named as IgA, IgD, IgE, IgG, and IgM. In human, IgA and IgG isotope can be further subdivided into closely related subclasses or subtype called IgA1, IgA2, IgG1, IgG2, IgG3, and IgG4. Mice, which are often used in the study of immune response, differ from human in that the IgG isotope is divided into IgG1, IgG2A, IgG2B, and IgG3 subclasses. Certain strain of mice, including the C57BL6, lack the gene of IgA2 but produce a related isotope called Ig2C. The heavy chain C region of all antibody molecules of one isotope or subtype have essentially the same amino acid sequence. This sequence is different in antibodies of other isotope or subtype. Heavy chains are designed by letter of the Greek alphabet corresponding to the isotopes of antibody. This is IgA containing alpha 1 gene, IgA2 alpha 2, IgD delta, IgE epsilon, IgG1 alpha 1, IgG2 alpha 2, IgG3 alpha 3, IgG4 alpha 4, IgM mu. In human IgM and IgE antibody, the constant region contains 4 tandem IgM domains. The constant region of IgG, IgA, and IgD contain only 3 IgM domains. These domains are genetically designed constant heavy domains and are numbered sequentially from amino acid terminals to carboxyl terminals. This is CH1, CH2, and so on. In each isotope, these regions may be designed more specifically. Different isotopes and subtype of antibody perform different effector function. The reason for this is that most of the effector function of antibodies are mediated by the binding of heavy chain constant region to Fc receptor on different receptor on different cells such as phagocyte, natural killer cells, and mast cell, and to plasma proteins such as complementary protein. Antibody isotopes and subtype differ in their regions, in their C constant region, and therefore in what they bind to and what effector function they perform. The effector function mediated by each antibody isotopes are listed in table 5,2 and are discussed in more detail later in chapter 13. Antibody molecules are flexible, permitting them to bind different array of antigen. Every antibody contains at least 2 antigen binding sites, each formed by a pair of variable heavy chain and variable light domains. Many Ig molecules can orient these binding sites so that the 2 antigen molecules on a planar Ig cell's surface may be engaged at once. This flexibility is conferred in large part by a hing region located between the constant heavy region 1 and constant heavy region 2 in certain isotopes. The hing region varies in length from 10 to more than 60 amino acid residue in different isotopes. Portion of this sequence assume an unfolded and flexible conformation, permitting molecular motion between the constant heavy chain 1 and constant heavy chain 2 domains. Some of the greatest difference between the constant region of IgG subclasses are concentrated in a hinge. This leads to different overall shapes of IgG subtypes. In addition, some flexibility of antibody molecule is due to the ability of each variable heavy domain to rotate with respect to adjacent constant heavy domains. So these are the human antibody isotopes. You saw the IgA, the IgD, the IgE, and IgG. So with their subclasses. The IgA is mostly in mucosal immunity. The IgD mostly in B cell antigen receptor. IgE defense against helminth parasite, immediate hypersensitivity. IgG opsonized complement activation, antibody-dependent mediated cytotoxic, and neonatal immunity because can be transferred from mother to child. IgM, this is pentamer, mostly in a naive B cell antigen receptor monomeric form. Complement activation, the main function of this antibody. There are two classes or isotopes of light chain called kappa and lambda chain that have distinct carboxyl terminal constant C region. Each antibody molecule has either two identical kappa light chain or two identical lambda light chain, but never one of each. In human, about 60% antibody molecule have a kappa light chain and about 40 have the lambda light chains. Marked changes in this ratio can occur in patients with B cells tumor because the many neoplastic cell being derived from one B cell clone produce a single species of antibody molecule, all with the same light chain. In fact, an abnormal predominance of either kappa-bearing cells or lambda-bearing cell is often used clinically for diagnosis of B-cell lymphoma. In mice, kappa-containing antibodies are about 10 times more abundant than kappa-containing antibodies, lambda-containing antibodies. Unlike in heavy chain isotopes, there are no known difference in function between kappa-containing antibodies and alpha and lambda-containing antibodies. Secretory and membrane-associated antibody differ in amino acid sequence of carboxyl terminal end of heavy chain region. The secreted form found in blood or mucosal secretions and other extracellular fluid contains a carboxyl terminal hydrophilic region called the tail piece. The membrane-bound form of antibody containing carboxyl terminal stretch that include two segments, a hydrophobic alpha helical transmembrane region followed by intracellular tail, which contain a juxtamembrane positively charged region of 3 amino acids. The positively charged amino acid bind to negatively charged phospholipid head group of inner leaflet of the plasma membrane and help anchor the protein into the membrane and sometimes called the stop transfer sequence. In the membrane, IgM and IgD molecules, the cytoplasmic portion of the heavy chain is short, only 3 amino acid residue in length. In membrane IgG and IgE molecule, the tail is approximately 30 amino acid residue in length and contain a significant motif that contribute to the memory B cell activation. So widely spaced surface determinant, you see, but closely spaced surface determinant. This is the same antibody binding to two determinants that are close together. This flexibility is mainly due to the hang joint. So the hang joint, you can see them, this widely spaced cell surface determinant. Secretory IgG and IgE and all membrane IgM molecule, regardless of isotopes, are monomeric with respect to the basic antibody structural unit. They contain 2 heavy chain and 2 light chain. In contrast, the secreted form of IgM and IgA form multimeric complex in which 2 or more of the 4 chain core antibody structural unit are covalently joined. IgM is secreted mainly as pentamer but also some hexamer of the core 4 chain structure, where IgA is usually secreted as a dimer. These complexes are formed by interaction between the tail pieces that are located in the carboxyl end of the secreted form of mu and alpha chain. Multimeric IgM and IgA molecules contain additional non-Ig 15 kilodalton polypeptide called the joining. This is J chain, which is disulfide bonded to tail pieces of Ig constant region and serves to stabilize the multimeric complex and to transport multilayer across epithelial cell from a basolateral to terminal ends. As we'll see later, multimeric form of antibody bind to antigen more avidly than monomeric form. Antibodies of different species differ from each other in C region and in framework part of the V regions. Therefore, when Ig molecules from one species are introduced into another, e.g. host serum antibodies or mouse monoclonal antibody injected to human, the recipient sees them as foreign, mount an immune response, and make antibody largely against the C region of the induced Ig. The negative impact of this anti-G response on use of antibody as therapeutic agent is discussed in more detail later in the chapter. Smaller sequence of differences are present in antibodies from different individuals, even of the same species, reflecting inherited polymorphism in the gene encoding C region of Ig heavy and light chain. When polymorphic forms of an immunoglobulin found only in some individual of the species can be recognized by antibodies, and the sequence that differ among individuals are called the allotypes. An antibody that recognizes an allotypic variation is called an anti-allotypic antibody. Even in the same individual, difference among different antibodies that are concentrated in the CDR constant isotypes of antibodies. An antibody that recognizes some aspect of the CDR of another antibody is therefore called anti-idiotypic antibody. There have been interesting theories that individuals produce anti-idiotypic antibodies against their own antibodies that can control immune response, but there is little evidence to support the importance of this potential mechanism of immune regulation. So we have the structure here, the membrane IgM and IgD. So you see these ones, the V region, variable region, these are transmembrane region, these are tail. And the green ones, these are light chains, light chain C. This color is stop transfer sequence. This color is a crustal tail, whereby you see only in mostly you see it in IgG, the crustal tail. You have the heavy chain C region, and you don't see the stop transfers to some other cells, some other IgG. So membrane and secreted form of immunoglobulin heavy chains. The membrane form of Ig heavy chains, but not on secreted form, contain transmembrane region made up of hydrophobic amino acid residue and the cytoplasmic domain that differ significantly among different isotopes. The cytoplasmic portion of the membrane form a mu chain and delta chain contain only 3 residues, whereas the cytoplasmic region of IgG and IgE membrane heavy chain, membrane alpha and Ilson heavy chain contain 20 to 30 residues. The secretory form of antibody and C-terminal tail pieces, which also differ among isotopes. Monoclonal antibodies. Monoclonal antibodies are a collection of identical antibodies. All molecules of a monoclonal antibody are produced by progens of a single B-cell clone, and all of the molecules therefore have the same variable region and bind to the same antigen that originally triggered the B cell. A tumor of plasma cell, myeloma, and plasmacytoma, like most tumor of any cellular region, is monoclonal and therefore produces antibody of a single specificity. In most cases, the specificity of the tumor-derived antibody is not known, so the myeloma antibody cannot be used to detect or to bind to molecule of interest. However, the discovery of monoclonal antibody produced by this tumor led to the idea that it may be possible to produce similar monoclonal antibodies of any desired specificity by immortalizing individual antibody-secreting cells from an animal immunized with a known antigen. A technique to accomplish this was described by George Kohler and Cesar Milstein in 1975, and this has proved to be one of the most valuable advances in biology and medicine. The method relies on fusing B cell from an immunized animal, typically a mouse, with with an immortal myeloma cell line, and growing the cell under conditions in which only the fused normal and tumor cell can survive. The resultant fused cells that grow out are called the hybridomas because they are hybrid of normal cells and the myeloma tumor. Each hybridomas makes only one Ig, derived from a B cell from immunized animal. The antibody secreted by many hybridoma clones are screened for binding to the antigen of interest, and clone with desired specificity is selected and expanded. The product of these individual clones are monoclonal antibodies, and each monoclonal antibody is specific for single epitope on the antigen used to immunize the animal. Monoclonal antibodies have many applications in research and in medical diagnosis and therapy. Some of their common applications include 1. Identification of phenotypic marker unique to particular cell types. The basis of the modern classification of lymphocyte and other leukocyte is the recognition of an individual cell population by specific monoclonal antibodies, most often by using flow cytometry and related technology or technique. See the appendix. These antibodies have been used to define cluster of differentiation, marker for various types. See Chapter 2 and Appendix 1. Immunodiagnosis. This is the second application, immunodiagnosis. The diagnosis of many infectious and systemic disease and even pregnancy relies on the detection of particular antigen or antibodies in the blood, urine, or tissue by the use of monoclonal antibodies in immunoassay. Tumor identification. Labeled monoclonal antibodies specific for various cell protein are used to determine tissue source of tumor by staining histological tumor section. Therapy. This is the fourth application. Therapy. Advances in medical research has led to the identification of cell and molecules that are involved in the pathogenesis of many diseases. Monoclonal antibody, because of their exquisite specificity, provide a means of targeting these cells and molecules. Many monoclonal antibodies are used therapeutically today. Some examples include antibody specificity for cytokine tumor necrosis factor, used to treat the rheumatic arthritis and other inflammatory diseases. Antigen against CD20 for treatment of B cell derived tumor, and depletion and for depleting a B cell in certain autoimmune disorder. Antibody specific for the T cell regulatory molecule PD1 and CTLA for used in therapy for many types of cancer. Antibody that bind to the epidermal growth factor receptor to target cancer cell. Antibody against the vesicular endothelial growth factor, a cytokine that promotes angiogenesis in patients with the macular degeneration and so on. The fifth function is functional analysis of cell surface and secreted molecules. In biological research, monoclonal antibody that bind to the cell surface molecule and either stimulate or inhibit particular cellular function are invaluable tools for defining the function of this molecule, including the receptor for antigens. Monoclonal antibodies are also widely used to purify selected cell population from complex mixture to facilitate the analysis of properties and function of these cells and block or deplete secreted molecules and particular cell for studying their function. One of the limitation of monoclonal antibodies for therapy is that these antibodies are mostly easily produced by immunized mice, but patients treated with mouse antibody will make antibody against the mouse IgG called human anti-mouse antibody. These anti-IgG antibody block the function and enhance clearance of injected monoclonal antibodies and also cause serum sickness. So see chapter 19. Genetic engineered technique has been used to replace the mouse sequence in the monoclonal antibody with the human sequence and thereby avoid anti-G response. The complementary DNA, copy of DNA that encode the peptide chains of monoclonal antibody can be isolated from hybridoma, and these genes can be manipulated in vitro. As discussed earlier, only small portion of antibody molecules are responsible for binding to antigen. The remainder of the antibody molecule can be thought of as framework. This structural organization allows the DNA segment encoding the antigen binding site from the mouse monoclonal antibody to be inserted into the copy of DNA encoding the human myeloma protein, creating a hybrid gene. When it is expressed, the resultant protein, which retains the antigen specificity of original mouse monoclonal, but has core structure of human IgG, is referred to the humanized antibody. Full human monoclonal antibody are also in clinical use. These are derived using phage display method or in mice with B cell expressing human Ig transgenes. The phage display technology for the generating a monoclonal antibody against the specific target involved the screening of bacteriophage libraries in which each bacteriophage displays on its surface one synthetically created antigen binding site to human Ig. By screening millions of such synthetic antibody binding sites for binding to antigen of interest, one that is specific for target can be and one that is specific target antigen can be identified. So the diagram, you see, isolate a spleen cell from the mouse immunized with antigen X. This is antigen X has been immunized. Then after you isolated a mixture of spleen cells, including some producing X antibody, then there will be a mixture fusion mixture. Mutant myeloma lines unable to grow in heart selection medium does not produce antibody, so you make it fuse. You see there is component of this, there is component of this. Then they are selected those one which in vitro selection in heart medium. You see this one are selected. Then only fused cell hybridoma grow. They grow. Then they grow a hybridoma producing monoclonal and X antibody are producing. So that is the process that is happening actually in the body. So that was just the descriptions. Example of monoclonal antibody in clinical use. We have alpha 4 integrins blocking immune cell ingress to intestine and CNS. This is used for treatment of Crohn's disease and multiple sclerosis. We have CD20. Effect is the depletion of B cell. They are used in rheumatic arthritis, multiple sclerosis, and other autoimmune disease, the B cell lymphoma. We have IgE, which is blocking IgE function. They are used in allergic-related asthma. We have tumor necrosis factor, reducing inflammation, the effect, and they are used treating rheumatic arthritis, Crohn's disease, and psoriasis. We have interleukin 17, reducing inflammation, used it for treatment of psoriasis. Apart from that, we have CD5, blocking the complementary mediated lysis, and they are used to treat paroxysmal nocturnal hemoglobinuria, atypical hemolytic uremic syndrome. Glycoprotein is inhibitor of protein platelet aggregation used for cardiovascular diseases. Rank ligands blocking the rank signaling. It's used for postmenopausal osteoporosis, bone metastasis of solid tumor, and SARS-CoV-2 proteins blocking the viral entry. They are used for treatment of COVID-19. So about cancer, we'll see it in chapter 18. So humanized antibodies are far less likely than mouse monoclonal to appear foreign in human and to induce antibody response. However, a proportion of subjects receiving full immunized monoclonal antibody for the therapy develop blocking antibodies known as human anti-human antibodies. HAHAs. Why this happen? it is in some individuals not understood. Synthesis, assembly, and expression of immunoglobulin molecules. Immunoglobulin heavy and light chain, like most secretory and membrane proteins, are synthesized on membrane-bound ribosomes in rough endoplasmic reticulum. The protein is translocated into the endoplasmic reticulum, and Ig heavy chain are N-glycosylated during the translocation process. The proper folding of Ig heavy chain and their assembly with the light chain are regulated by protein resident in the endoplasmic reticulum called chaperones. These proteins, which include calnex and molecular called binding proteins, bind to newly synthesized Ig peptide and ensure that they are retaining or targeting for degradations unless they are they fold properly and assemble into complete Ig molecule. The covalent association of heavy and light chain is stabilized by formation of disulfide bond, which also occur in endoplasmic reticulum during the assembly process. After assembly, the Ig molecules are released from the chaperones, transported to the cysteine of the Golgi complex, where carbohydrates are modified and then routed to the plasma membrane in vesicle. The antibodies of the membrane forms are anchored to the plasma membrane and secreted into the transported out of the cell. The maturation of B cell from bone marrow progenitor is accompanied by specific changes in Ig gene expression, resulting in the production of Ig molecule in different forms. The earliest cells in the B lymphocyte lineage that produces Ig pulpetad called the pre-B cells synthesize the membrane form of the mu chain, heavy chain. These mu chains associated with a protein called surrogate light chain to form the pre-B cell receptor, which is expressed on the cell surface and provides signals that promote maturation. Immature and mature B cell produce kappa and lambda light chain, which associate with a mu protein to form IgM molecule. Mature B cells expresses membrane form of IgM and IgD, the mu and delta heavy chain associated with kappa and lambda light chain. These membrane Ig receptors serve as cell surface receptor that recognize antigen and initiate the process of B cell activation. The pre-B cell receptor and B cell antigen receptor are monovalently associated with two other integral membrane proteins, IgA alpha and Ig beta, which serve signaling function and are essential for surface expression of IgM and IgD. We will discuss the molecule of cellular events and cellular event in B cell maturation underlying these changes in antibody expression in chapter 8. When mature B cells are activated by antigen and other stimuli, the cell differentiates into plasmablast and then into antibody-secreting plasma cell. This process is accompanied by changes in pattern of Ig production. One such change is increased production of the secreted form of Ig relative to the membrane form. This alteration occurs at the level of post-transcriptional process. The second change is expressing of Ig heavy chain isotype other than IgM, IgD by a process called heavy chain isotopes, the class switching. A third change involves the introduction of new amino acid substitution into the variable domains of antibody heavy light chain to create high affinity antibody resulting into the changes in antibody that is called affinity maturation. Changes in antibody expression that occur after B cell activation will be discussed later in this chapter and more in detail in chapter 12. So this was the immunoglobulin. This is the diagram figure. Stage of maturation. We have the stem cells, pattern of immunoglobulin production none. Then it comes to the pre-B cell, whereby the cytoplasmic mu heavy chains and pre-B receptors. Then immature B cell, membrane has IgM now. Then in mature B cell membrane has IgM and IgD. Activated B cell, the plasmablast, low rate of Ig secretion, heavy chain isotope switching, affinity maturation happen here. Then we get the plasma cell. This is one with high rate of Ig secretion, little or no membrane Ig. Half-life of antibodies. The half-life of circulating antibody is the measure of how long those antibodies remain in blood after secretion from B cell or after injection in case of administered antibody. The half-life is the mean time before the number of antibody molecules is reduced to half. Different antibody isotopes have very different half-lives in circulation. IgE has very short half-life, about 2 days in circulation. Although cell-bound IgE associated with high affinity IgE receptor on mast cell has a very long half-life. See chapter 20. Circulating IgA has half-life of 3 days. Although most IgA is produced at the mucosa site and is secreted directly into the lumen of the gut airway, the circulating IgM has half-life of about 4 days. In contrast, circulating IgG molecule has half-life of about 21 to 28 days. The long half-life of IgG is attributed to its ability to bind to specific Fc receptor called Neonatal Fc Receptor, so called because it is involved in the transport of IgA from the maternal circulation into the fetus. The Neonatal Fc Receptor structure resembles the MHC class I molecule described in chapter 6. It is expressed in the placenta where it transports the IgG molecule from the maternal blood across the cell and into the fetal circulation. See the chapter 14. In adult vertebrate, the neonatal Fc receptor is found on endothelial cell, macrophage, and other cell types, and bind to IgG that is ingested from the blood by pycnosis into endosomes. The IgG remain bound to FcR in the acidic environment of the endosomes. Fc does not target the bound IgG to lysosome, the usual fate of many ingested molecule, but recyclate it to the cell surface and release it to the neutral pH of blood, returning the IgG to their circulation. The intracellular sequestration of IgG away from lysosome prevents IgG from being degraded as rapidly as most other plasma protein, including other antibody isotopes, and as a result of IgG isotope has relatively long half-life. There are some differences in the half-lives of 4 human IgG subtype. IgG3 is relatively short-lived because it binds poorly to the FcRn. IgG2 and IgG1, IgG2, and IgG4 are long-lived. The long half-life of IgG has been used to provide a therapeutic advantage for certain injected protein by producing fusion proteins containing biologically active part of proteins and Fc portion of IgG. The Fc portion enables the protein to bind to Fc and thus extend the half-lives of injected proteins. One therapeutic useful Fc fusion protein is TNFR1J, which consists of extracellular domain of the type 1 tumor necrosis factor receptor fused on IgG Fc domain. Tumor necrosis factor receptor 1 bind to tumor necrosis factor and block the inflammatory action, similar to anti-tumor necrosis factor antibody, and it is used to treat certain immunological disorder, i.e. rheumatic arthritis, inflammatory bowel disease, and psoriasis. Another Fc fusion protein used therapeutically is CTLA, containing extracellular domains of a CTL4 receptor, which bind to and block B7 costimulator fused to Fc fusion of human IgG. It has also been used to treatment of rheumatic arthritis and kidney transplant rejection. So this is the neonatal Fc receptor contribute to the long half-life of immunoglobulin IgG. We continue. The antibody binding of antigen. All of the function of antibody depend on the ability to specifically bind antigen. We will now consider the nature of antigen and how they are recognized by antibodies. Features of biologic antigens. An antigen is any substance that may be specifically bound by antibody molecule, the T cell receptor. The antigens that are recognized by antibodies may be almost every kind of biological molecule, including simple intermediary metabolites, sugar, lipids, alkaloids, hormones, as well as molecules such as complex carbohydrate, phospholipid, nucleic acid, and protein. This is in contrast to most T cells, which only recognize peptide. Not all antigens recognized by specific lymphocyte or by secreted antibody are capable of activating lymphocyte. Molecules that stimulate immune response are called immunogen. Macromolecules are effective at stimulating B lymphocyte to initiate humoral immune response because B cell activation require the bringing together, cross-linking of multiple antigen receptor. A small chemical such as dinitrofen may bind to antibodies and therefore antigen, but they cannot activate B cell on their own because they can bind to only one B cell receptor at a time. To generate antibodies specific for such a small chemical, immunologists commonly attach multiple copies of small molecule to a protein or polypeptide before immunization. And in this case, a small chemical is called hapten, and the large molecule to which it is conjugated is called carrier. The hapten carrier complex, unlike free hapten, can bind to the multiple antigen receptor on B cell and hence act as immunogen. This phenomenon has been explored to produce effective vaccine. Macromolecules such as protein, polysaccharide, and nucleic acid are usually much larger than the antigen binding region of an antibody molecules. Therefore, any antibody binds to only a portion of the macromolecules, which is called determinant or an epitope. These two words are synonymous and are used interchangeably throughout this book. The portion of the antibody that directly interacts with the epitope has been called an paratope. Macromolecules typically contain multiple determinants, some of which may be repeated and each of which by definition can be bound by antibodies. The presence of multiple identical determinants in an antigen is referred to the polyvalence or multivalence. Most globular proteins do not contain multiple identical determinants and are not individually polyvalent, but many identical proteins may be displayed in polyvalent array on cell surface, including the surface microbes. In the case of polysaccharide and nucleic acid, many identical epitopes may be regularly spaced and repeated in the same molecule, and these molecules are said to be polyvalent. Polyvalent array of carbohydrate antigen also can be displayed on surface. Polyvalent antigen can induce clustering of B cell receptor and thus initiate the process of B cell activation. So we look to this, the presence of polyvalence. This is the presence of multiple identical determinant in antigen referred to as polyvalence or multivalence. And we have this, what is called the repeated molecules, either carbohydrate or amino acid, which is called polyvalent. The spatial arrangement of different epitopes on single protein molecule may influence the binding of antibody in several ways. So these are the conformational epitopes. You see maturation, epitope host, linear epitopes, so different appearance. So when when determinants are well separated, two or more antibody molecules can be bound to the same protein antigen without influencing each other. Such determinants are said to be non-overlapping. When two determinants are close to each other, the binding of antibody to the first determinant may cause steric interference with binding of antibody to second. Such determinants are said to be overlapping. In rare cases, the binding of one antibody may cause the conformational change in the structure of antigen, positively or negatively influencing the binding of a second antibody or at another site on protein by means other than steric hindrance. Such interactions are called the allosteric effect. Any available shape or surface on a molecule that is recognized by antibody constitutes an antigenic determinant or epitope. Antigenic determinant may be present on any type of molecule, including but not restricted to the carbohydrate, protein, lipid, and nucleic acid. In case of protein, the conformation of some epitope depends only on the primary structure, and conformation of other determinants reflect tertiary structure or conformational changes. Epitope formed by several adjacent amino acid residues are called the linear epitope. The antigen binding site for antibody can usually accommodate a linear epitope made up of 6 amino acids. If linear epitope appear on external surface or in region of extended conformation in native folded protein, they may be accessible to antibody. In other cases, linear epitope may be inaccessible in a native conformation and appear only when protein is denatured. In contrast, the conformational epitopes are formed by amino acid residues that are not in sequence but become spatially juxtaposed in the folded protein. Antibody specificity for certain linear epitopes and antibody specificity for conformational epitope can be used to ascertain whether the protein is denatured or in its native conformation, respectively. Protein may be subjected to modifications such as glycosylation, phosphorylation, ubiquitination, acetylation, and proteolysis. These modifications, by altering the structure of protein, can produce epitope. The new epitope also may be generated in tumor by mutation of gene encoding self-protein. Such epitope induced by post-translational modification or by mutation are called the neoantigenic epitope, and they too may be recognized by specific antibodies. Structural and chemical bases of antigen bindings. The antigen binding site of many antibodies are planar surface that can accommodate conformational epitope of macromolecules, allowing the antibody to bind larger molecules. The 60 CDRs, 3 from the heavy chain and 3 from the light chain, can spread out to form a broad surface. In number of antibody specificity for small molecules such as monosaccharide and the drug, the antigen is bound into the cleft generated by close apposition of CDRs from variable light chain and variable heavy chain. The recognition of antigen by antibody involves non-covalent reversible binding. Various types of non-covalent interaction may contribute to the antibody binding of antigen, including electron forces, hydrogen bond, van der Waals forces, and hydrophobic interaction. The relative importance of each of these depend on the structure of binding site of individual antibody and the force of antigenic determinant. The strength of binding between single combined site of antibody and an epitope of antigen is called the affinity of antibody. The affinity is commonly represented by dissociation constant, which indicate how easy it is to dissociate an antigen-antibody complex into the constant by changing their concentration. A small dissociation constant indicates a stronger or high affinity interaction because A low concentration of antigen and of the antibody is required for complex formation. The dissociation constant of antibody produced in typical humoral immune response usually varies from 10 power 7 10 power negative 7 to 10 power negative 11. Serum from an immunized individual will contain a mixture of antibody with different affinity for antigen. Because the hing region of antibodies give them flexibility, and the single antibody may attack to a single multivalent antigen by more than one binding site. For IgG and IgE, this attachment can involve at most two binding sites, one on each Fab. For pentameric IgM, however, a single antibody may bind to up to 10 different sites. Polyvalent antigen will have more than one copy of particular determinants. Although the affinity of any one antigen binding site will be the same for each epitope of polyvalent antigen, the strength of attachment of antibody to antigen must take into account all the binding site of antibody and all the available epitopes of antigen. This overall strength of attachment is called avidity and is much greater than the affinity of any one antigen site. Thus, low affinity IgM molecule can still bind tightly to polyvalent antigen because many low affinity interaction up to 10 per IgM molecule can produce high avidity interaction. Polyvalent antigen are important from the viewpoint of B cell activation, as discussed earlier. Polyvalent interaction between antigens and antibody are also of biological significance because many effector functions of antibody are triggered optimally when two or more antibody molecules are brought close together by binding to polyvalent antigen. If polyvalent antigen is mixed with a specific antibody in a test tube, two interact to form immune complex, and the correct concentration called the zone of equivalence, antibody and antigen form an extensively cross-linked network of attached molecules such that most or all of the antigen and antibody molecules are complexed into large masses. Immune complex may be dissociated into small aggregates either by increasing the concentration of antigen so that free antigen molecule will displace antigen bond to antibody, zone of antigen excess, or by increasing antibody so that free antibody molecules will disperse bound antibody from antigen determining zone of antibody excess. If zone of equivalence is reached in vivo, larger immune complex can form in circulation. Immune complexes that are trapped on or formed in the walls of blood vessel can initiate inflammatory reaction, including resulting in immune complex diseases. See Chapter 9. Structure, function, relationship in antibody molecules. So, related to this, let us finish this one, the structure. So, the structure function relationship in antibody molecule. Many structure features of antibody are critical for the ability to recognize antigen and their effector function. In the following section, we will summarize how structure of antibody contribute to their function. Features related to the antigen recognition. The ability of antibody to specifically recognize a wide variety of antigen with varied affinity reflects the properties of the variable region. So, we have specificity. Antibody can be remarkably specific for antigen, distinguishing between small differences in chemical structure. The fine specificity of antibody applies to recognition of all classes of molecule. For example, antibody can distinguish between two linear protein determinants differing by only single conservative amino acid substitution that has little effect on secondary structure. This high degree of specificity is necessary so that antibody generated in response to an antigen of one microbe usually do not react with the structural similar self molecule of which the antigen of other microbes. However, some antibody produced against one antigen may bind to different but structural related antigen. This is referred to the cross reaction. Antibodies that are produced in the response to microbial antigen sometimes cross-react with the self-antigen, and this may be the basis of certain diseases like you will see in chapter 19. Diversity. As we discussed earlier in this chapter, an individual is capable of making a tremendous number of structural distinct antibodies on the order of millions, each with distinct specificity. The ability of antibodies in an individual to specifically bind to a large number of different antigens is the reflection of antibody diversity. And the total collection of antibody with different specificity represent antibody repertoire. The genetic mechanism that generates such a large antibody repertoire are active only in B lymphocyte, and the same mechanism for generating tau-like receptor diversity are active in T cells. The diversity is generated by a random recombination of a limited set of inherited germline DNA sequence to form a functional gene that encodes for variable region of heavy and light chain, as well as by addition of nucleotide sequence during the recombination process. We will discuss this mechanism in detail in chapter 8. The millions of resulting variation in structure and are concentrated in antigen binding hypervariable region of both heavy and light chain, and thereby determine specificity for antigen. Sorry for moving too fast. So this is we have this one, the IgG, the structure, the valence, avidity. If you have monovalent, it's low. If you have bivalent, like IgG, it's high. If you have polyvalent, like IgM, it's more high. So that is called the issue of diversity. We see zone of equivalence. They are similar antigen antibody complex. The size of antigen antibody immune complex are functional of relative concentration of antigen antibody. Large complexes are formed by concentration of multivalent antigen and antibodies that are termed the zone of equivalence, and the complexes are smaller in relative antigen or antibody excess. This here we are speaking of changes in antibody structure during humoral immune response. Illustration depicts the changes in structure of antibody that may be produced by Progenitor activated B cell, one clone, and related to the change in function. During affinity maturation, mutation in V region, indicated in yellow, can happen. Yeah, so you can read more in detail about that. Another character that defines this antibody is affinity maturation. The ability of antibody to neutralize toxin and infection microbe is dependent on tight binding of antibodies. As we have discussed, tight binding is achieved by high affinity and high avidity interaction. A mechanism for the generation of high affinity antibodies involves subtle changes in structure of variable region of antibody during humoral immune response to protein antigen. These changes can come about by process of somatic mutation in antigen stimulated B lymphocyte that generate a new V variable domain structure, some of which bind the antigen with greater affinity than the original variable region domain. Those B cell producing higher affinity antibody preferentially bind to some antigen, and as a result of selection become a dominant B cell with each subsequent exposure to antigen. This process is called affinity maturation. The result is an increase in average binding affinity of antibody for antigen as humoral immune response evolve. Thus, an antibody produced during a primary immune response to a protein antibody often has a KD in average of 10 power 7 to 10 power 9. In subsequent response, the same antigen secondary response as may occur in repeated interaction with some species of microbes or repeated immunization. The affinity increases often resulting to KD 10 power 11 or even less. We will discuss mechanism of finite maturation in chapter 12. Feature related to the effector function. Many of the effector function of antibody are mediated by Fc portion of molecule, and Ig isotope that differ in this Fc region perform distinct function. We have mentioned previously that effector function of antibody require the binding of heavy chain C region, which make up Fc portion to other cells and plasma protein. For example, IgG coats microbes and target them for phagocytosis by neutrophil and macrophage. This occurs because IgG molecule is able to simultaneously bind through its Fab region to microbes and through its Fc region to IgG heavy chains, specific receptors that are expressed on neutrophil and macrophage. In contrast, IgE bind to the mast cell and trigger their degranulation because mast cell expresses the IgE specific Fc receptor. Another Fc-dependent effect of mechanism of humoral immunity is activation of classical pathway of complement system. The system generates inflammatory mediator and promote microbial phagocytosis and lysis. It is initiated by binding complement protein called C1q to Fc portion of antigen complex IgG or IgM. The Fc receptor binding and complement binding site of antibody are found within heavy chain of C domain of the different isotopes. We will discuss the structural and function of Fc receptor and complementary protein in chapter 13. The effector function of antibodies are initiated only by Ig molecules that have bound antigen, not by free IgG. The reason that only antibody with bound antigen activate effector mechanism is that two or more adjacent antibody Fc portion are needed to bind to and trigger the various effector system, such as complement protein and Fc receptor phagocyte. This is a requirement for adjacent antibody molecules ensure that effector function are targeted specifically toward eliminating antigens that are recognized by the antibody, and that circulating free antibody do not inappropriately or dangerously trigger the effector response. Changes in isotopes of antibody during humoral immune response influence how the response works to eradicate antigen. After stimulation of an antigen, a single clone B cell may produce antibodies with different isotopes that nevertheless possess identical V domain and therefore identical antigen specificity. Naive B cell simultaneously produce IgM and IgD that function as membrane receptor for antigen. When these B cells are activated by foreign antigen, typically of microbial region, they may undergo a process called isotope class switching, in which the type of constant heavy region and therefore the antibody isotope produced by B cell changes, but the V region and the specificity do not. As a result, isotope switching, different progen of the original IgM and the IgD expressing naive cells may produce isotope and subtypes that are best able to eliminate the antigen. For example, antibody response to many bacteria and viruses in the blood is dominated by Gg antibody, but the same microbes in mucosa tissue, intestine, and airway elicit much of IgA, which is efficiently secreted into the lumen of this organ. Switching to IgG isotope also prolong the effectiveness of humoral immune response because of the long half-life of IgG antibodies. We will discuss the mechanism of function of significance of isotope switching in chapter 12. The heavy chain C region of antibody also determine the tissue distribution of antibody molecules. As discussed earlier, after B cells are activated, they gradually lose expression of the membrane-bound antibody and express more of its secreted protein. IgA can be secreted efficiently across mucosal epithelium, and it is the major class of antibody in mucosal secretions and milk. Neonates are protected from infection by IgG antibodies. They require they acquire from their mother through the placenta during gestation. This transfer of maternal IgG is mediated by FcRn, which was described earlier as receptor responsible for long half-life of IgG antibodies. Summary. Antibodies or immunoglobulin IgEs are a family of glycoprotein produced in membrane-bound or secreted by B lymphocyte. Membrane-bound antibodies serve as a receptor that mediates the antigen-triggered activation of B cell. Secreted antibody function as mediator of specific humoral immunity by neutralizing microbes and toxin and by engaging various effector mechanisms that serve to eliminate the bound antigen. The antigen binding region of antibody molecules are highly variable, and any one individual has potential to produce millions of different antibodies, each with distinct antigen specificity. All antibodies have a common synthetic core structure of two identical covalent linked heavy chain and two identical light chain, each linked to one of heavy chain. Each chain consists of two or more independently folded IgE domain of about 110 amino acid containing conserved sequence of intrachain disulfide bonds. The N-terminal domain of heavy and light chain form the V region of antibody molecule, which differ among antibodies of different specificities. The V region of heavy and light chain each contain three separate hypervariable regions of about 10 amino acids that are specially assembled to form antigen combining site of antibody molecule. Antibodies are classified in different isotopes and subtypes on the basis of difference in a heavy chain constant region, which consists of 3 to 4 IgC domains, and these classes and subclasses have different functional properties. The antibody classes are called IgM, IgD, IgG, IgE, and IgA. But light chain of single IgE molecule are of same isotype, either kappa or lambda, which differ in their single constant domain. Most of the effector function of antibody are mediated by C region of the heavy chain, but these functions are triggered by binding of antigen to the combining site in the variable region. Monoclonal antibodies are produced from single clone of B cell and recognize single antigenic determinant. Monoclonal antibodies can be generated in laboratory and are widely used in research, diagnosis, and therapy. Antigens are substance specifically bound to antibodies or T lymphocyte antigen receptor. Antigens that bind to antibody include a wide variety of biological molecules, including sugar, lipid, carbohydrate, protein, nucleic acid. This is in contrast to the most T-cell antigen receptor, which recognize only peptide antigen. Macromolecule antigen containing multiple epitopes or determinants, each of which may be recognized by antibody. Linear epitope of protein containing consists of sequence of adjacent amino acid and conformational determinant are formed by folding the propeptide chain. The affinity of interaction between the combining site of single antibody molecule and the single epitope is generally represented by KD, calculating from binding data. Polyvalent antigen contain multiple identical epitope to which identical antibody molecule can bind. Antibody can bind to, or in case of MG, up to 10 identical epitope simultaneously, leading to enhance the avidity of antibody antigen interaction. The relative concentration of polyvalent antigen and antibody may favor the formation of immune complex that may deposit in tissue and cause damage. Antibody binding to antigen can be highly specific, distinguishing small difference in chemical structure, but cross-reaction may also occur in which two or more antigen may be bound by same antibody. Several changes in structure of antibody made by one clone of B cell may occur in cause of an immune response. B cell initially produce only membrane-bound IgE, but inactivated B cell and plasma cell, IgE with same antibody binding specificity as original membrane-bound IgE receptor is secreted. Changes in use of C constant region gene segment without changes in variable are the basis of isotype switching, which lead to the changes in effector function without a change in specificity. Point mutation in variable region of antibodies specificity for an antigen leads to the increased affinity for that antigen, affinity maturation. So these are selected reading, indicate publication of historical interest, generally reporting the discovery of phenomenal process that has later shown to be fundamental. This you'll see this one. Many, but not all, of these discoveries lead to the Nobel Prizes of discoveries. Nature of discovery is summarized briefly in each reference. So you will see these are the discoveries that shed light in immunology that are important for us to remember. So I think you are a little bit tired. These are the references, and let's get prepared for the next chapter. We see chapter 6, antigen presentation to T lymphocyte and the function of major histocompatibility complex. I think it's more interesting as you are moving to adaptive immunity. It was a little bit difficult in innate immunity, but as we go up, we are learning some skills that will help us to grow and shed more light. Thank you and all the best. Jeremiah Puza.